Thanks everyone for joining. I'm really excited to be here and to share some of the recent literature that has been published with Metabolon data. And I'm just excited to share it all with you guys so you can hear about all of the advancements that are being made in drug discovery applications utilizing metabolomics. So what is metabolomics? Metabolomics is the study of small molecules. So any of the small molecules that are present in a system is where that realm of metabolomics lies. So we're thinking about, all of the precursors and products of all of the biochemical processes that happen in a system. And that can encompass sugars and amino acids and lipids and fatty acids and all of those, smalls and abiotics. Really anything that's smaller than 2,000 Daltons is something that is considered a small molecule and falls under the umbrella of metabolomics. And that encompasses, like I mentioned, all of those biochemical pathways within a system. When we start to think about glycolysis and gluconeogenesis and TCA cycle and sort of all of those pathways that we're, all of those metabolic pathways when we're thinking about energy production and storage and catabolism, that's where metabolomics lies. And it encompasses all of those reactions that are revolving around that. So it can be binding and dissociation, degradation, transport of these small molecules. Like I said, the precursors and products of some of these enzymatic reactions. And metabolomics is what captures all of those pieces of information. And it really is closest to the phenotype and captures that snapshot of metabolism of what's happening within a system. It falls within the hierarchy of sort of the central dogma of biology. So if we're thinking about genomics and transcriptomics and proteomics, if we're thinking about that replication and transcription and translation piece of the puzzle, metabolomics really does fall on that hierarchy because these are all of those intermediaries in between those processes. So the genomics piece of the puzzle is really what could be. The transcriptome is sort of the activities and what's happening. And then when we get down to the proteomics and the metabolomics side, we're really looking at the functional expression of what is actually happening. And like I said, that snapshot of metabolism and that snapshot of biology is where metabolomics works best. So diving right in, I wanted to share with you guys today a paper called The Discovery of DRP-104, a tumor-targeted metabolic inhibitor prodrug. This was from a group out of Johns Hopkins, and they wanted to investigate the capabilities of this drug. So it is known that glutamine metabolism is altered in tumors. It's widely well known. The understanding has become a significant focus in cancer research over the past few decades. Glutamine metabolism has implications in carbon and nitrogen storage and catabolism and other important metabolic intermediates related to energy. It also impacts redox homeostasis via oxidative stress. So it really has a huge implication with cancer metabolism and tumor metabolism in that tumor microenvironment. DON, 6-diazo-5-oxo-L-norleucine, DON, forever here called DON, is a selective irreversible inhibitor of enzymes that utilize glutamine. And you can see the similarity between the molecules here. So you can see the similarity between the glutamine and the DON molecule and how that is obviously a very competitive inhibitor of glutamine using enzymes or molecules. Although DON is a really successful competitive inhibitor of cancer tumors or cancer cells, effectively killing tumors, it has some good T cell effectivity. It has off-target effects in the GI tract and other tissues with high glutamine demand. So it really sort of was ineffectual as a cancer treatment because of these off-target effects. So this group out of Johns Hopkins wanted to investigate if having a prodrug form of DON would be a better solution. And they know that because tumors are protease rich, they wanted to utilize this a prodrug form of DON by putting two moieties onto the DON molecule, an isopropyl ester, and then also a carboxylate and an acetylated tryptophan on the amine. So this molecule, DRP-104, isopropyl 5-2-acetometo 1H indole 3-ill-propionamide 6-diazo-5-oxohexanoate, forever now called DRP-104, was developed, and they wanted to move forward with this prodrug version of DAWN to test for the safety, efficacy, and dosing in humans, human cells, and mouse models. So some of the first work that this group investigated was how this DRP-104 was persisting in both mouse and human plasma. We can see that the DRP-104 was stable and present in both the mouse model and the human plasma through their testing to look at the stability of that. And you can see that it's present in their mouse models and the human cell lines in the plasma. It's not released into the GI, so it wasn't really detected in the GI tract and it wasn't stable there. But then the original drug, that DAWN, was released into the GI tract in both the mouse model and the human cell line. So in order to ensure that the DRP-104 was also just as effective against cancer as DAWN, they did a cell viability study. They showed that the treatment with either DAWN or DRP-104 was cytotoxic and that the M1 metabolite was not cytotoxic. So they were able to demonstrate that. Of note, they also utilized FGAR, an intermediate in the de novo purine synthesis pathway that is known to be altered by DAWN as a measurement of tumor engagement. And this was important because it's a precursor in the purine synthesis pathway, and it affects the production of ADP, inorganic phosphate, and glutamate. So they wanted to use that as an indicator of target engagement. They administered DRP-104 and DAWN to mice with flank tumors, and where they also harvested the tissue in order to determine where DRP, DAWN, and the M1 metabolite were localizing. So you can see here on those measurements, they were able to show that the DRP-104 was stable for up to and present for up to 2 hours in the plasma, but not persisting in the tumor or in the GI tract. The DAWN was present in the plasma, the tumor, and the GI tract, and then that inactive form of DRP, the M1 metabolite, was present in the GI tract. And so they were able to show that the M1 metabolite was localizing. And you can see that the DRP-104 was present and stable, and where it was located and up in the GI tract for up to 5 hours. They also investigated the efficacy of DRP-104 against other types of cancers, and they were able to demonstrate its effectiveness against colon, breast, and lung cancers as well. By exploiting the protease inhibitors present in the non-tumor tissues, the chemistry itself of DRP-104 helped ensure the safety and specificity of this drug. So you can see here on the diagram that DRP-104 undergoes an amide hydrolysis to DAWN isopropyl ester, and then it's hydrolyzed to the DAWN drug impact itself in the intestinal matrix. But you can see that the M1 metabolite is stable and there's no spontaneous hydrolyzation of the M1 metabolite to the DAWN drug form. So they were able to really characterize this drug just based on its very simple chemistry, and they were able to show that it's very specific to specific tissues and it's very effective. So they were able to demonstrate that DRP-104 was effective against not only tumor size, but the overall weight of the mouse. So you can see here that when the mice that with tumors were treated with the vehicle versus the DRP-104 or the DAWN, that the tumor size in the vehicle treated mice still continued to increase, while in the DRP-104 and the DAWN treated mice, there was no growth of that tumor. And you can also see that the weight of the mice was affected by the treatment, whether it was the vehicle, the DRP-104, or the DAWN-treated mice. You can see here that the weight of the DAWN-treated mice was actually being reduced, and so they were actually losing weight over time, which was affecting their physiology and their overall health of the mice. And you can see that that affected the survival rate. So in the vehicle-treated mice, they were dying off much faster than in the DAWN-treated mice. They had slightly longer survival rates, but because of that physiology that was being affected, they weren't surviving as well as when they were treated with that DRP-104 because of that effect of the DAWN on the GI tract. So you can see here on the histology plots where when they were treated with the vehicle, there's no effect on the GI histology and the cellular structure of the GI tract. But when they're treated with the DON, you can see the inclusions into the bacterial mat. And when they're treated with the DRP-104, there's much less of that effect, and it looks closer to the vehicle-treated mice GI tract. So they also wanted to utilize this same approach and the same analyses to optimize their dosing strategy for DRP-104. They did a subcutaneous administration of DRP-104 and showed that it had no adverse effect on the GI histopathology at the lower doses. So you can see here at that 0.1 milligram per kilogram and the 0.3 milligram per kilogram, there was no effect of the DRP-104 on the mice and their GI tract. And when we get up to the 1 milligram per kilogram treatment, you start to see a little bit of that effect, but it's not nearly as bad as even with the treatment with the DON. And they were able to show that the treatment with these different dosages of the DRP-104 were still effective against tumor size without affecting the overall weight and physiology of the mice themselves. So a lot of careful thought and observation went into determining the effectiveness and dose of DRP-104, but to truly understand the mechanisms, metabolomics was performed. So utilizing Metabolon's untargeted global platform, 400 annotated metabolites were detected. You can see clear differentiation between the DRP-104 and the vehicle-treated mice on the principal component analysis. So you can see here there's two clear separations between our DRP-104, which are our red points on the PCA plot, and then between our vehicle, which are the green points on the PCA plot. And you can see there's quite a significant amount of separation between these two groups at 43.3% on PC1 and 11% on PC2. There were 88 differentially abundant metabolites by t-test, and they were able to show that the major changes between these two groups were present in energy metabolism, nucleotide, and amino acid metabolism. And they were really able to narrow in on to what was happening in this tumor microenvironment at that metabolic level. So DRP-104 treatment demonstrated significant effects on multiple TCA cycle intermediates. You can see the increased amounts of glutamine detected in DRP-treated cells and subsequent downstream glutamine metabolites lower in the DRP-104 treated mice. So you can see those significantly increased amounts of glutamine in the DRP-104 treated mice, and then you can start to see where some of those intermediates are lower, and then again when we get to that fumarate and some of those more downstream molecules, they're significantly different. So purine, not only was the TCA cycle affected, but purine and tryptophan metabolism was also decreased in DRP treated mice. And this is significant due to that purine FGR relationship that we were talking about earlier. So the FGR-AR is important in that de novo purine synthesis pathway, and it increased with DRP-104 treatment. And so the products of the purine synthesis, including adenine and guanine, were decreased upon the DRP-104 treatment. And this is pretty indicative because when we're looking at some of those changes between the purine metabolism, and then they also saw changes in tryptophan and kynurenine metabolism, this is all really indicative of an immunosuppressive response. And that supports the earlier findings that the glutamine antagonism affects indoleamine metabolism, which is a mechanism that is really relevant to its efficacy when combined with immunotherapy. So in order to confirm the TCA metabolites that were down, because they were seeing some of that trending in the global metabolomics, they also did some flux analysis to look at those downstream molecules from glutamine. And you can see again, some of those downstream molecules were significantly different when we're looking at the treatments after the DRP-104. Because Metabolomics confirmed these major shifts in metabolism, the researchers moved on to investigate additional treatment strategies. So they knew that anti-PD-1 treatments and DRP-104 each individually were very effective against tumor size without affecting the weight and the physiology of the mice. But because those changes that they saw in the tryptophan and kynurenine pathway, that indicated that a combination therapy might be more effective because of that immunosuppressive response. And so they were able to show that indeed, when the mice were treated with both, they had significant survival rates. So you can see when they're treated here with both the anti-PD-1, the DRP-104 individually, or the combination of the two, they had a much more significantly increased lifespan. So you can see here in black that survival rate is all the way to the end of their treatment, whereas without any treatment at all, their tumor sizes improved and they passed away much faster. And then with each individual treatment, it just wasn't as effective as that combination treatment. The mice were treated and cured with the 2 weeks of the treatment of the DRP-104 and the monotherapy. But then they also did a 60-day washout, and they did a re-challenge with the tumor, and they were able to show that there was no uptake and no proliferation of those cancerous cells after their treatment. And so not only is the DRP and the PD-1 combination effective in treating the cancer, it was also effective in preventing the cancer from coming back after that 60-day washout. They also looked at the tumor infiltrating CD8 plus T cells in DRP-104 treated mice, and they were able to show increased expression of activation markers CD44, CD69, and TBET, as well as markers of stem-like memory phenotype TCF1, CD28, BCL6, EOMES, CD122, CD127, and CD62L. So, all in all, they were really able to demonstrate that the anti-tumor response triggered by the DRP-104 was dependent on the ability of the drug to enhance the endogenous immune response of the CD8 mediated mechanism. And they were really able to show that there was this metabolic reprogramming, and that glutamine blockade was affecting the tumor microenvironment metabolism. So all in all, they were able to really demonstrate that DRP-104 is a really promising prodrug alternative to glutamine inhibiting DON. It was sustained in the tumor, which they were able to demonstrate. It was minimized off-target effects that they were able to show with their histopathology. It was effective against tumor proliferation and re-challenge after a 60-day washout period. And it also enhanced the CD8 plus T cell mechanisms and enhanced that memory phenotype. Metabolomics was really valuable to this study because it was able to show the divergent metabolism of DRP-104 in the tumors versus the normal tissue, and it enabled that selective targeting of cancer cells, potentially improving the therapeutic index and reducing side effects. So they were really able to show that metabolic shift in amino acid, nucleotide, and TCA metabolism. They were able to validate the use of their tumor engagement biomarker, their FGAR, by looking at the purine base accumulation that was measured by metabolomics. They were able to go towards a combination therapy because of the changes they were seeing in the tryptophan and kynurenine pathway as well. So the metabolomics really informed their treatment strategy, and glutamine metabolic changes and downstream TCA intermediate abundances were greatly affected, which is what they predicted would happen, and they knew would happen because of the form of DON, but they were able to validate all of those findings through the metabolomics. So pushing right along, just to talk about Metabolon and metabolomics a little bit, what we do and what we can offer. So Metabolon has been around for over 20 years. We have 185 employees. We have experts in mass spectrometry, data curation, bioinformatics. We have a lot of biologists on staff that will help you interpret your data. And all of these people and experts that we have on our team, and all of the time that we spent curating our product and our data and making sure that it's really high quality and the highest level of fidelity has made us really the leaders in the field of metabolomics. Thinking about what our capabilities are for just pharmaceutical-based research specifically, we've done over 500 projects with just pharmaceutical-based, clinical trials and research. We work in all different model species, so humans, non-human primates, murines, canine cell models, sort of any model system that seems to make sense for your research. We work in all different types of sample types. What you think about really standard for, a human health focus, whole blood, plasma, serum, saliva, feces, sebum, but we also will work in, any pretty exotic sample types as well. So this isn't necessarily specific to just pharmaceutical research. Metabolomics can be widely applied, but if we're just thinking specifically about pharmaceuticals, we work in multiple different therapeutic areas: gene therapies, cell therapies, biologic, drug delivery. We've been published in hundreds of clinical trials, and we are CAP, CLIA, and ISO certified as well. We really like to be your partner in your research as well. So, we really like to be involved early and often in those conversations about metabolomics as you're starting to plan those. Don't hesitate to reach out to us and have a conversation started with us, because we really want to be here to help you think about what's the most bang for your buck when you're doing metabolomics, what's the best way to design your study, what makes the most sense, and what's going to be answering the right questions. What are we asking the right questions, and how can we answer those? We can give you advice about sample collection. We'll do all of the sample preparation, so that's nothing that you have to worry about. Again, I talked about our team of experts. Our mass specs experts have developed 4 different individual LC-MS methods that are each individually optimized to cast the widest net and capture as many molecules as possible in our untargeted approach. Then we have our team of curation scientists, like I mentioned as well. So we're able to curate, those thousands of data points from the data that we've collected from your samples, and we're able to curate that against our library, where we're able to make metabolite annotations, and then we're able to interpret the results of that and look at the pathways and really parse out the story of the biology that's occurring in those samples. There's 2 major buckets of what we do here at Metabolon. We have our discovery side. So when we're thinking about traditional untargeted metabolomics and a global approach, when we're casting a wide net, we're trying to see as many molecules as possible. We have some smaller discovery panels and some targeted analyses as well. So we have some large targeted analyses where we're casting a pretty wide net, like our complex lipid targeted panel, where we're looking at 1,100 lipids. And then we can be really granular, and we can look at individual metabolites as well. And some of these are off the shelf, so please feel free to take a look at our website. Like I said, shoot us a question. We're always happy to chat. So when we're talking about our global and targeted analysis, in our library, we have over 5,400 annotated metabolites. And so what that means is that not only at our level 1 validation, 80% of those metabolites are at level 1 validation. So what that means is that not only have we collected all of the pieces of orthogonal data, retention time, exact mass, fragmentation patterns, spectral pattern, we've collected all of those pieces of data, but then for 80% of those metabolites, we've also been able to run a neat standard on our platform. So if we've been able to acquire or synthesize a neat standard, we've been able to really validate that the metabolite that we're talking about is actually the metabolite. And so we're really talking about that biochemistry, and we really can make a lot of insights about to what's actually happening in that system. There is a small amount of metabolites in our library that are at level 2, and that means that we have not been able to acquire or purchase a neat standard for those metabolites, but we still have all those pieces of orthogonal data, so we're still able to make a highly competent identification for those metabolites. We just haven't been able to run that neat standard. So really, we're still able to speak to the biology that's happening and really talk about the changes that are happening. And it's not sort of this it might be this molecule, there's 20 potential identifications for this molecule, that type of thing. When we're talking about putative IDs, we're really confident in the library and the identifications that we're able to annotate and make. Metabolon offers, a few different levels of deliverables, but coming standard with any deliverable that you get is our client data table, which includes a heat map of all of the metabolites that we detected in your samples. They're categorized by subpathway and superpathway, and they're representative in a heat map. They're represented in a heat map format. So we do some standard statistics, t-tests, ANOVAs, and COVAs, and then we're able to represent the changes that are happening within your biological groups as a heat map. So fold change up and down, and statistical significance represented as red and green. And that can be really helpful when you're looking at whole pathways and looking for changes that are happening across a whole pathway, for example. We also offer individual line or bar plots of every metabolite. And that can be really helpful when you're building out figures or, making reports or writing a manuscript. You can also have a CSV of your data file. So you can work with the data however you like. It's your data. You can treat it and process it however you would like to do that. And whatever statistics you might want to run that are additional or different to what we offer, you certainly have that capability. We have some bioinformatics tools that are really exciting that were just released this year. I'll talk about those in just 1 minute, but not to be diminished in any way is our biological interpretation. That's where one of our translational scientists parses through your data. They look at the changes that are happening in all the different pathways, and they interpret what the implications of that might be. So they help you write that roadmap of the biology that's happening. They look at the crosstalk between the pathways, and they're really able to help you flesh out that biological story. So like I mentioned, there's new and exciting bioinformatics tools that we launched just this year within our platform. So this is also with any project that you would do at Metabolon, you get access to these multivariate analysis tools and these visualization tools, which again makes you writing your manuscript and getting your data out there and your research out there for public consumption really straightforward and streamlined. Principal component analysis, partial least squares discriminant analysis, PLSDA, volcano plots, clustering and hierarchical clustering with heat map representation, pathway analysis. And then what's really innovative to Metabolon is our Lenses Explorer, where you're able to look at your data through the lens of a specific biological pathway or a disease, or if there's specific metabolites that you're really interested in because you've read a piece of literature and you're like, this set of metabolites might have implications within my research, you can specifically filter your data so you're just looking at the results of your global metabolomics with those specific pathways enabled. So it can be a really efficient way and a really unique way to look at your data from a different angle. I mentioned some of our targeted panels as well. I'll talk briefly about our complex lipids targeted panel. It looks at over 1,100 lipids in 14 different classes of lipid species. Complex lipids, it's exactly what it sounds like. We're analyzing complex lipids. So they can be really difficult to look at. There's different head groups, there's different tails, there's different levels of saturation. There's a lot of isobars of these different lipids. And, it can be a really challenging class of molecules to look at. So with our solution, we've really been able to make the most of leveraging ion mobility separation by getting the most out of each identification of those lipids by running a single point calibration. We run 50 internal standards, so we're able to quantify those lipids and give you a molar concentration of all of the lipid classes or all of the lipid species that we detect. And then like I mentioned, it is very comprehensive in coverage. It has 14 lipid classes with 1,100 lipid species on it. So it's a really nice solution if you're looking at, thinking about some of those long, nonpolar, neutral, complex lipid species, tags, DAGs, cholesterol esters, those types of molecules. We have 22 off-the-shelf targeted metabolite panels. Please feel free to check out our website, email us with any inquiries you might have. These are targeted assays where you're able to get absolute quantitation of the molecules that are included on these different individual panels. They can be really beneficial when you're looking for that absolute nanogram per milligram type of concentration of specific molecules of interest in your research. And then, not to be diminished, I like to talk about our level of validation that we have within our lab. We're an ISO 9001 certified lab. The implications of this cannot be diminished in any way. We hold ourselves to the very highest standards of sample handling and reporting your data. It means that we have an SOP for all of the processes that are involved in accepting your samples and processing your samples and reporting the data back to you and how we do that. So we really hold ourselves to a really high level of data validity with you. We also have some of our targeted assays that are GCP and CLIA and CAP certified as well. The next piece of literature that I wanted to talk to you about is called Integrated Multiomics, reveals glucose use reprogramming and identifies a novel hexokinase in alcoholic hepatitis. This is from a group out of UNC Chapel Hill, Thomas Jefferson University, University of Pittsburgh, and a lot of others. There was a lot of co-authors on this paper. There was obviously a lot of work that went into this. So it's just a really nice piece of research that I was excited to share with y'all today. Alcoholic hepatitis is a severe form of liver disease, and it has a really low survival rate. So stats on exactly how many people will develop this disease annually are vague, but it is important to understand these extreme mechanisms because it is such a fatal disease to have. It's poorly characterized, and there's really limited treatment options. And these researchers really recognize the need for improving those treatment options and better characterizing alcoholic hepatitis. They were able to analyze tissues from control patients, alcoholic hepatitis patients, alcoholic cirrhosis, AC tissues, and they utilized a metabolomics and an RNA-seq approach to be able to do so. They really focused their work on a global metabolomics approach. There were 869 metabolites detected across the 3 different tissues. And you can see there's clear differentiation on our PCA plot between those different tissue types. So our normal tissue, our AC tissue, and our AH tissue with some significant differentiation on PC1 at 26.14% and PC2 at 12.81%. Statistical pairwise comparisons show that some differentiated metabolites were shared between these comparisons. So you can see there was 113 that were shared when we compared, or when they compared AC versus control, AH versus AC, and AH versus control. So they were able to show that some of these were shared differences between all of these comparisons, but that there were also some unique ones as well. And their AH phenotype was the most differentiated, which was the one that they were most significantly interested in. And they were able to show that across those 58 metabolites, pathways like amino acid metabolism, carbohydrate, vitamin, energy metabolism, and nucleotide metabolism were all affected in this AH phenotype. They wanted to take a deeper look at the carbohydrate metabolism as it was paramount to characterizing the AC and the AH phenotype. They were able to show that across central carbon metabolism, AH liver had a unique profile. So they were able to really show what was different in the AH versus the control across all of the central carbon metabolism. So all the way down through glycolysis and the TCA cycle, they were able to show these differences. And by taking this closer look at glycolysis, Metabolomics showed that glucose 6-phosphate was 200% higher in the AH phenotype, and hepatic glucose, 3-phosphoglycerate, phosphoenolpyruvate, and those types of molecules were all significantly decreased in the AH phenotype compared to the control or even the AC phenotype. Citrate and aconitate, the first 2 products in the TCA cycle, so we can see here, were also increased in our AH phenotype and by 170 and 380% respectively compared to the control group. And then they were able to also do their RNA-seq analysis, which confirmed the changes here. And so you can see the RNA-seq results here on the enzymatic reactions, and you can see where they are agreeing and aligning with a lot of the metabolomics results here that are represented. They were also able to show that a novel hexokinase that converts glucose to glucose 6-phosphate, similar to HK1 and GCK, was 600% higher in AH tissue compared to control. So just to take a closer look at that reaction, so this novel hexokinase HKDC1 was significantly increased in the AH patients, and we can see where that's affecting our glucose 6-phosphate as being much more or much higher amounts. Our glucose was down in our AH versus control, and we can start to parse out where these changes are happening in this metabolic pathway by integrating the metabolomics and the transcriptomics results. They were really able to characterize that the AH liver was incapable of proper glucose usage and storing. You can see that it's getting shunted to the pentose phosphate pathway immediately. And these changes were really contributing to the lack of liver regeneration. RNA-seq alone wouldn't have characterized these changes at a granular enough level to demonstrate where the metabolic bottleneck was occurring. As you can see, there's changes in these enzymes all the way down, starting to go through that beginning, those first step or those earlier middle steps of glycolysis. But taken together with the metabolomics results, they were able to see that in the alcoholic hepatitis patients, the pentose phosphate shunt was being heavily utilized, and that this major shift in metabolism was likely responsible for the liver failure and the high mortality of AH disease. In addition to integrated omics analysis, they also did correlation analysis. They did pairwise Spearman rank correlation tests between each gene and metabolite, which demonstrated correlations in the AH tissue. You can see succinate in particularly has a really strong inverse correlation with TCA cycle relevant genes, again, suggesting the presence of a bottleneck in the mitochondrial Krebs cycle. All of these changes are indicative of altered storage and release mechanisms of these energetic pathways, and metabolite sinks and bottlenecks within the central carbon metabolism. So you can see some of these relationships with these TCA cycle intermediates with the transcript omics data was really able to help solidify these relationships and this metabolic shift that was occurring. So they wanted to dive into HKDC1 more since it was so much more abundant in this AH phenotype. It's a homologous protein to HK1, hexokinase 1, and it was a really good predictor of short-term mortality in these patients. So you can see where it's indicative of this decreased survival. So high amounts of HKDC1 resulted in a shorter survival time. You can also see the much higher levels of HKDC1 in the hepatic tissue compared to the other tissues and compared to the other disease types. It was also indicative of acute kidney injury as well. So they were able to show that with that 32-fold change cutoff threshold. They were then able to validate this in the serum levels as well. So you can see again that AH levels was much higher than the control and the other types of liver injury and liver disease in the serum levels, as well as the liver tissue. Again, the association with acute injury was associated in the plasma and in this confirmation cohort as well. And again, the high levels of the HKDC1 were indicative of a lower survival time than with the lower levels of HKDC1. So really, the presence in the strong AH phenotype of the HKDC1 and the decreased survival indicate that this could be an effective biomarker for clinical treatments. And the relationships between kidney injury and mortality and the relationship with HKDC1, again, identify it as a biomarker for clinical measures and survival. This was really the first study to characterize AH liver tissue at this level. So they were able to characterize the changes in central carbon metabolism, glycolysis, gluconeogenesis, TCA cycle, compared to the AC phenotype and the control. The integrated transcriptomics and metabolomics really revealed the energy metabolism shift that was occurring in the AH patients. So they were really able to show the bottlenecks, the dysregulated glucose storage, and the pentose phosphate shunt that was occurring. HKDC1 was identified as the most upregulated kinase in the AH patients. Those high levels of HKDC1 were really associated with acute kidney injury and decreased survival rates. So overall, Metabolomics revealed this extensive reprogramming of glucose metabolism in alcoholic hepatitis, and it really identified HKDC1 as a novel therapeutic target and biomarker for this disease. And this metabolic shift wouldn't have been properly characterized without the use of metabolomics. The transcriptomic data really identified that there was issues in the TCA cycle and in glycolysis, but the changes were occurring further downstream than at this specific bottleneck point of HKDC1, that transition from glucose to glucose 6-phosphate. So without the metabolomics, they wouldn't have been able to narrow in their target quite as precisely if they had just used the RNA-seq data. So, hopefully today we've talked about all of the ways that metabolomics could inform your drug discovery pipeline. We can be involved in basic research and discovery. We can be involved in preclinical and clinical trials. And then, we can also, metabolomics can be utilized for a post hoc toxicity and efficacy type of approach as well. So metabolomics is really widely applicable in the pharma space and in the drug discovery pipeline. So hopefully we've been able to demonstrate that for you today. I was really excited to share all of this research with everyone. So please don't hesitate to ask any questions in the chat, or we can provide resources or anything that might be helpful when you're thinking about doing metabolomics and thinking about how Metabolon might be able to help you. So hopefully we've been able to demonstrate some of that for you today. I appreciate your time and thank you so much for listening.